Hello everyone, and thank you for joining us. I'm here with Dr. K, and we are going to be doing some fun science lab. Go ahead and share in the comments if you were joining us live or if you were joining us on the replay. I'm excited to hear where you guys are joining us from, and if you guys have any questions, be sure to share them in the comments, and Dr. K will be able to answer them for you guys. Would you be willing to tell us a little bit about yourself and your business?
Hi, I'm Dr. K, and I both tutor and teach homeschool classes. In 2020, I started teaching on OutSchool, and since then, I found so many passionate young scientists, and I've really loved connecting with them and teaching labs and teaching science classes. And so today, I'm going to do a little bit of what you might see in one of our chemistry or science clubs. It's not quite a full lab, but it is a very fun experiment. Is it okay if I share my screen?
Absolutely.
Okay, perfect. Let's see. I'm going to put in the chat, I'm going to put in, I have a little bit of a handout if you would like to follow along after, or I'm sorry, not follow along. If you would like to do this afterwards, I have a little bit of a write-up for it, so I'm going to put that in the chat. It's just a PDF, and afterwards it has all of the information about the lab that we'll be doing today.
Hi, Jennifer. Thanks for joining us. We have your son that is 10 and your daughter that's 6 that's joining today.
Perfect. Okay, are you able to see my screen?
You just put that up on the screen there. There we go.
Perfect. Hi, everyone. I'm going to be talking about something called chromatography today. And we're going to do a bit of an experiment. I have some of my materials. I will talk about as we get going too, but essentially I have some strips of paper towel, a little bit of water, and some washable markers. And I'm going to talk about why we need all of these different things in order to do this experiment today. So a little bit about myself. I am a research chemist. I do a lot of research with something in chemistry that deals with looking at the mass of certain compounds. I have been a research chemist for about 10 years. I have taught a variety of chemistry classes from middle school all the way up to graduate level chemistry. I went into chemistry not really knowing much about it in high school and college, and really fell in love with science. You might be able to guess based on this if you've seen, I'm going to turn on my little pen. If you've seen Finding Nemo, you might recognize this. I am from Australia. Ooh, I don't know what happened with my pen there, but I'm from Australia. So this is the Sydney Opera House, and a lot whenever I put this in the slide, someone says just like in Finding Nemo. But before we get started, all students will get a free session of our Science Sleuth Club. This was going to start back up again the first week of October. So this runs year round. We do take about 2 or 3 week breaks throughout the year around different holidays, around the start of the semester, just to give students some time to get acclimated back to their classes. But it will start up the first week in October. I believe our first middle school science club experiment is called Finding the DNA, or extracting DNA in strawberries. We generally do science experiments that are fun, and we relate it back to a real world application. So for example, we use strawberries and extract DNA. We also do some slime because slime is so much fun to make, and there's a lot of chemistry behind making slime. We also learn a scientific principle each week. So even though we do a very fun experiment, just like today, we're going to learn a scientific principle. Now, sometimes the words are big and sometimes the scientific principles seem complicated, but when we really break it down to something fun like chromatography or DNA extraction, it's easier to apply. So in the chat, I put the link to the handout for this week, and then I have some information here. I put my website for Hypatia STEM Academy, and in this case, we meet weekly Thursdays at 4:30 p.m. EST each week, and this is open for everyone from 5th through 8th grade science. But I'm going to talk a little bit about our experiment today. So for our lab, we're going to be talking a lot about colors, a lot about dyes, Sometimes I get questions about dyes versus pigments. So generally dyes are going to be what is in a liquid. So we're going to be talking about dyes today. Now, we might have learned about primary colors in other classes. Our primary colors are red, yellow, and blue. I have a little picture over here. Let's see if I can get where we have red. It's actually looking a little pink now that I look at it, yellow, and blue. In this case, we can use these to make different colors. We can use red and yellow to make orange. We can use blue and yellow to make green, and we can use blue and red to make purple. Now, in these cases, if you've ever tried to mix paint together, you might notice that you can make different shades of orange, purple, and green if you add more red, or if you add more yellow, or if you add more blue. Now, colors and markers can be a bit more complex. In this case, marker makers or the manufacturers like Crayola and RoseArt and all of those different marker makers, you might notice that if you use a Crayola marker, your red might be a little bit different than if you use a different brand's red. The reason for this is because there are thousands of dyes that make up what we think of as red, and all of these are different chemical compounds. Now, in this case, if we have all of these different dyes that make up red, you can imagine that sometimes we have different combinations that make up something like orange. Sometimes even, and I'll show you this today, sometimes red and orange and yellow and things like that are also made up of multiple dyes to make up those primary colors. So even though they're primary colors, sometimes marker manufacturers put in multiple dyes to make even a red color. And we'll see that in our experiment. Now you might wonder, or you might have seen some markers say washable or ultra clean or ultra washable. So what does that mean? Now I have 2 kids, and so all of the markers in my house are generally washable markers, and if they're not, they're generally put up very, very high in my house. And what washable means is that they can be washed out with water. So washable is really just saying that if you rinse something with water, it will come out. And so in the case of washable markers, so I have an example here. I have a black Crayola that says an ultra clean washable, and it says ultra washable. In this case, if I accidentally drew on my shirt with this, theoretically, if I put it in the laundry and washed it in the washer with water, the water should be able to get this black pen out. But if I were to accidentally draw on my t-shirt with a black Sharpie, now a Sharpie is a permanent marker. This does not wash out with water because the dyes in here are not similar to water. And so there are things that would get out Sharpie, but they're not water. They're different liquids. With this black one, this washable one, I'll bring it back. The dyes in here are very similar to water in some properties, and so that's why it can come out with water. And we're going to be using this today. We're going to be using that we know that it'll come out with water in order to do our experiment. So we might be wondering, can we separate out, if there's more than one dye in our marker, can we separate out the different dyes? Can we separate out to see what might be in there? And the answer is yes. We can use a technique called chromatography. Now, chromatography is a technique to use to separate mixtures. It's used to separate a lot of things, not just dyes. It's used all throughout science. The word chromatography comes from the Greek chroma, meaning color, and graphene, meaning writing. So in this case, this is color writing. Now in chromatography, we can use it to look at a variety of things. Now, the prettiest ones are things with colors, but in science, and I'll talk about this on the next slide, sometimes things don't have colors. So what do we do? Now, you might have heard of some other words that are derived from graphene, things like calligraphy, choreography, photography. If you have any words you can think of that might have the same ending, would you mind putting them in the chat So if you can think of any other words that end with that graphy, that graphy, like chromatography, calligraphy. So calligraphy is that beautiful writing. Choreography is like the writing of dance steps. Photography is sometimes considered the writing of light, so using that light to make photographs. So if you can think of any other words that you think might be derived from graphene and we can talk about, or if anyone can think of one that I didn't include in this list.
What about cinematography?
Perfect, yeah, cinematography. And so that's where we have the writing of film. That's a great one. So we can use these root words like graphene to figure out what some of these other words mean. And so even though chromatography, calligraphy, choreography, photography, cinematography are all very, very different, we have some of the same root words.
And Jennifer added oceanography.
Perfect. Yes, oceanography. We see that a lot when we're talking about learning about marine biology in the ocean. We have that where we are writing and writing things about the ocean. Geography is very similar to that one as well. Great job, everyone. So where do we use chromatography? As I said, this is great for colorful mixtures. Scientists use chromatography every day to separate different chemicals. So what I have here is a bit of a complex chromatography column. We won't be doing something like this today, but this is another form of what we're doing today. And you can see this is big. It's a little hard to see the scale, but this would probably be from here to here, about 2 or 3 feet tall, just to show the scale of that. So I've done chromatography multiple times in my career, and sometimes it's very colorful mixtures. Sometimes it can be green, especially when we're using things from plants. So you can separate the chlorophyll from other parts of the plants. If you were to crush up plant leaves and you were to crush up plant leaves and extract out some of the things in plants, you could separate the beta carotene and chlorophyll from each other using chromatography. You can also use this for foods, medicines, and ink. Now I said, what about if it's not colorful like this? What if we don't know if it's blue, light blue, green, or orange? What if this just all looks white? Some things fluoresce under a black light. So what you can do is you could look at this under a black light and you might see bands that have fluorescence or are glowing a different color. And sometimes that's what we have to do in chromatography if everything is colorless. So today ours is going to be very colorful, but we can still use chromatography as long as it either fluoresces or glows a certain color, or if one thing, our thing of interest, might be a certain color, we can use chromatography for a lot of things. Now today, we're going to be performing a technique called paper chromatography. Now, the one I showed before was a very big example. That's if you're wanting to separate large amounts of plants or medicines or inks, something like that. Now we just want to separate a tiny amount. And so we're going to be using something called paper chromatography. What we're going to do is we're going to have a strip of paper. In this case, you could use, I just have some strips cut out here. You could use, I have 2 different types of paper towel. You could use paper towel. You can cut it to different strip lengths. You just want a strip that's long enough for you to be able to hold at the top without getting your fingers wet. So you can cut it as big as you want. You just want to make sure that you can hold on to the top of that. Now with paper chromatography, what we have is that we're going to use washable markers, water, and that paper towel to see if we can separate the mixture of our dyes. What we're going to do is we're going to take our strip and we are going to put little dots of our washable marker on the bottom. So we're going to put those dots, and then what's going to happen is we're going to place this in water. And the water is going to start moving up our paper towel using something called capillary action. Now, sometimes my students have heard capillary action another place, and that is when we talk about plants. So when plants get water from their roots, the way they get it all the way up to their top is also capillary action. It's the same thing. So the same way plants get water all the way up from their roots is the same way that our water is going to travel up our paper towel through capillary action. So I'm going to go over how to do this experiment. So I'm going to stop sharing really quick so I can get my screen back, and then we'll come back to this PowerPoint because it does need to sit for a bit, and I'm going to have some fun trivia questions. So let me get back here. Okay, perfect. So what you're going to need, I'm going to show some different steps. I'm going to push this back. Back a bit, there we go. Okay, so what you're going to need is you're going to need some water. You don't actually need that much. And I'm using a clear beaker, but you can use any sort of glass or cup. The only thing is you're going to want to be able to see down in there. So what I mean is that you'll need to be able to see essentially into your paper towel and watch as it comes up. So what you're going to do is you're just going to pour a small amount. You don't actually need that much, just enough to cover the bottom. I'll talk about in a second why you don't want to put much in there. But we're going to take our paper towel. Now what I have here is just a paper towel, and then I've cut it into strips coming this way. So I've just taken scissors and I've cut up to make a strip. Now in this case, this is what we're going to be doing chromatography on. This is our paper for our paper chromatography. Now you're going to take whatever colors you want. I have a variety of pens. I have a lot. I've stolen them from my kids. I have all of their pens and their markers right here, and I can put some dots at the bottom. So what I'm going to do is I'm going to take my black marker, and I'm going to have to do, see if I can show this. I'm going to do it up on my hand. So I'm going to take it, and I don't want to put it at the bottom. I'm going to want to put it about an inch or so up from the bottom. I'm going to put a dot. I'm going to put it down here, and I'll show you. So I'm going to put a dot. And as you can see, I've put a little dot. You're going to want it a bit bigger than that. So you're going to want to dot, as you can see, I'm going like this. I want to get a lot of ink on there. Now, the reason I want to get a lot of ink, I'm just dotting about 10 times in that spot. And I'll show you at the end as I'm going through what will happen if you don't put a lot of ink. So I have some examples of what happens if we do certain things. So you want to put it a bit up from the bottom. The reason for this is we are going to put this bottom in the water. If the ink is where we put it in the water, all of that ink will also move out into our water, and we'll see that water turn very vibrant and colorful, but that's not what we want because it does affect our results a bit. So I'm going to do a couple of colors. I'm going to do black. I'm going to do red. So I'll do some dots here. I'll show you in a second. So really want to get a lot of ink on there in that one spot. They don't have to be very big. Let's see, where'd my purple go? You can pick whichever colors you want. I'm going to do 4 on mine. And what we're going to do is we're going to make something that'll look like a very colorful caterpillar. And what I like to do at the end is draw on a little face. Okay. So I've done 4 colors here. And what some of my students like to do as well is if you want to do it a second time after this, you can put 2 colors on top of each other to see if you can separate them. So what I'm going to do for this last one to show this, I have an orange right here. So I am going to put another color on top of this orange. So I am going to put, let me see what color I didn't do. I'm going to put green on top of this orange. And so what we should be able to do is see those 2 colors separate. Okay. So I have my paper. I have all of my dots. I've now put some green over top of this orange. And now what I'm going to do is I am going to put it into my beaker. And so what you're going to see as soon as I put this in here, you're going to see that capillary action move very fast. So let me make sure it's on the screen. So that water is going to start coming up. you should be able to start seeing it move. Here it goes. Those dye spots are already starting to move all the way up. And you can see, I'll give it a second and then I'll bring it closer. Let me see if I can, you can see that first spot, which is now, I guess, the last because my camera is mirrored. That last spot is black, and you might notice that it looks like there's two colors. So when I did this experiment, I noticed that my black pen, I thought it would be more than one color, but it looks like it's actually a red and a green mixed together. And so we'll see that as we let this separate more, but you can see over here, there is two colors coming out that black. So what I'm going to do is I'm going to let this sit. It does take a bit. If you want to, you can tape this. So what I sometimes do is get a thing of soda or tape it to a wall in order to let it continue. So I'm just going to let it sit right here. Just going to put this in here so that it can continue. Perfect. Okay. So while this is running, it does take, depending on the paper towel. So I've noticed that depending on the quality of the paper towel that I use, sometimes it takes a little bit longer, sometimes it doesn't take that long. So I'm going to let this run while we do a little bit of a trivia experiment, and then we'll come back and I'll show some of these experiments. So let me share my screen again. So I have some fun. Let's see. I have some fun facts or fun trivia about colors in chemistry. So they don't necessarily have to do with chromatography, but I have some fun facts about color. So my first quiz question, and if you want to answer it in the chat, what color does fabric turn when it's added to boiled avocado stones or pits? And so I have three options for colors: green, blue, or pink. And so I sometimes get students who have done this. It's a very fun experiment. You can boil avocado stones and dye fabrics like t-shirts or washcloths, and it'll turn a certain color. And so if you'd like to put in the chat your guess.
I'm going to guess pink.
Oh, perfect. Yep, we can guess pink.
Jennifer guesses pink as well, and they guesses green.
Perfect. So let's see. I have a picture of what color it turns, and it turns pink. Great job. What's interesting about adding avocado stones to water, they have a dye in them that when it's dyed on things like cotton, it turns pink, which is a really fun fact about those avocado stones. Perfect. Okay. I have one more. Purple dye was historically very rare and reserved for nobles. It was called Tyrian purple, and the reason it was so rare, it was very hard to extract from something. And do you think it was extracted from tree bark, sea snails, or spices? And so you can put in the chat what you think it might be extracted from.
Okay, I'd love to see you guys' guesses. We're just waiting here for them to come in the comments.
This one might be a tricky one.
Because you really can get dyes and things from all three of those things.
Yeah,
so Jennifer is guessing sea snails.
Perfect.
And Vay, do you guys have a guess? I'm also going to say sea snails.
Perfect. Okay,
and Vay says spices.
Perfect. Yes. So the answer is sea snails. So when this color was originally being extracted, this comes from sea snails. I have a little picture of what the actual chemical compound that they're extracting from. So these specific sea snails have this sort of compound up here. And depending on which type, you can get sort of any of these three colors I have in this picture from a museum. But these were hunted essentially so much that they almost went extinct. But in order to get 1 ounce of this dye, so a very, very tiny amount of this dye, you would need 20,000 sea snails. And so this has to do with how it was so rare. So in order to dye, you can imagine a cloak or some sort of shirt, you would need a lot of sea snails. And so that's why it was essentially reserved for nobles. It took a lot of work to get this dye. So
That's crazy. That's a lot of sea snails.
it is a lot of sea snails. And so let's see, I have a couple more. Okay. Copper is a metal that is used in many things, including pennies. So that's a US penny. When it oxidizes, it turns, and I have some example answers, green, blue, or red.
Just waiting for the answers to come in here in the comments. I'm going to say green.
Perfect. So we will vote for green.
Jennifer says red.
Perfect. So in this case, copper is a metal, and it's used not just in pennies. There's a lot of things that use copper. When it oxidizes, this is a chemical change. So oxidation can happen when it's just sitting around because, of course, there's oxygen all around us. And when copper oxidizes, like in pennies, it turns green. And one very big example of a huge oxidation experiment is the Statue of Liberty. So I have a picture up in the corner of what the Statue of Liberty looked like when it very first came to the US, or I think this is probably right before it came to the US. In this case, we can see it looks just like pennies. It looks that beautiful copper color. Now, as it oxidizes or as it has that chemical change, it turns more and more green. And that's how we have the Statue of Liberty we see today, which is very, very green. That's the oxidation.
Okay, for some reason I did not know that it wasn't like originally green. That is crazy.
Yeah, so you can actually see they've taken photos as, from the time the Statue of Liberty came, and you can see it get slowly more and more green to the green that we know today. So,
That is
yeah.
super cool.
Yeah, so this is just essentially all of that copper, the same thing that happens to pennies, how we sort of have this, if you look at old pennies that you might have in your wallet or floating around your house, that green on it is the same green that's on the Statue of Liberty. There are some other types of oxidation reactions that you might see around your house. One is if you leave iron nails or if your hammer has iron in it outside, it'll rust. And that rust is another type of oxidation. But in that case, instead of copper, that's iron. So iron is another metal. And if you leave nails out that have iron in them, they'll rust. So you might see, at construction yards, you'll see rusty nails on the ground. And then the third type that I always like to talk about is silverware used to be made almost exclusively of silver. And some of my students hear it in period movies or movies that are set a bit ago, maybe the early 1900s, and they would hear someone say, oh, you should go polish the silver. And sometimes I get students asking me, why do they need to polish the silver? And this is because of another oxidation. Silver tarnishes, and tarnishing is an oxidation, just like this copper turning green, but silver turns sort of a dull blackish color. It's sort of a brownish black color if you've ever seen silverware tarnish or silver any piece. So those are all oxidation reactions. Okay,
Jennifer says this experiment is super cool. My kids really like it, and the colors are turning out beautiful.
Oh, perfect. I am glad. So mine's drying right now, and then I will show in a second. It's almost done. So I have two more, and then I'll show you some of mine. Okay, our second to last one, I have two more, so this one and another one. What color is a polar bear's skin? White, black, or pink?
Oh, I don't know. I think it's white. What do you guys think? Just Jennifer says she's guessing black.
Perfect.
And Vay is also guessing black. So I'm being outvoted here.
So we have 3 options, white, black, or pink. It sounds like a lot of people are voting black. We have this cute little baby polar bear here. And in the case of polar bears, their skin is black. I found this very sad polar bear who had to get shaved for vet treatment to show this black skin up underneath. So their skin is black, but the reason it doesn't look black is their fur is actually hollow, and that reflection of light can make it look like it's white instead of black. So that's a fun little fact about those. And then I have our last trivia question. What color does tonic water glow under a black light or UV light? Green, blue, or red?
The fact that it glows at all is a super cool fact. I'm going to guess blue because that's what I want it to be.
Perfect. Yeah, so this does glow. Based on the question, it does glow. And I always encourage, if you do have a black light around your house, if you take some just tonic water that's at the store, even just a Canada Dry bottle, I just went on Google and found a picture. As long as it says contains quinine, this compound right here, it will glow. And it's actually that compound in it that glows in the tonic water. And I'll talk about why they include that compound once we talk about what color it glows.
So Jennifer says her kids are mixed. It's blue or red.
Perfect.
And Vay, what is your guess? I know we have a couple more people that are lurking, so you guys can guess too.
Yeah, so it does glow under a black light. So this isn't a trick question. It does glow. That would be mean to put a trick question. So what I'll do
Okay, Vay is also, the reason it took a while is they're also debating between blue and red as well.
Perfect. Okay, so I'll show you because I have a picture of what it looks like. And it glows blue under a UV light. And it is very blue. So if you do have a black light at home or you have access to a black light, you can go to the store. Tonic water is normally around $2. And it has this compound called quinine in it. And what it is quinine is actually a malaria treatment. And this was put in to help with people who had gotten malaria or who were worried about getting malaria, and they would drink tonic water. And so this is still in our tonic water, this compound. This is what quinine looks like. And when you have your tonic water and you put it under a black light, it's that quinine that's glowing blue. And so this is a great fun experiment if you do have a black light lying around. So that is my last trivia. So what I'm going to do is I'm going to stop sharing. I'm going to come back over here. Perfect. Okay, so mine is all done. It's still a little wet, but I'm going to turn it around. So just show what my colors were. You can see it's maybe a little hard to see on my screen, but there is a pink or red smudge all the way up until here, and then it turns into green. So this black pen that I have, this is my Crayola black pen, seems to be made up of a pink or red dye and a green dye. Now, I had said at the start of that presentation that sometimes even primary colors are made up of multiple dyes. It's a little hard to see here, but you see a really concentrated red spot here, and then another concentrated red spot up here. So what's probably happened is this red dot is made up of multiple red dyes. It looks like my purple is actually only made up of one dye. So even though we can talk about how purple is the mixture of red and blue, there are dyes that are just purple. And in this case, it looks like they've used for this purple pen one purple dye. And then you can kind of see it's a little hard to see. The green did separate out a little bit here from the yellow, which is sort of down here. It was starting to separate, but if I had let it go a little bit longer, it probably would have separated more. So I have a couple more examples. I had done this one. So this is some different colors. This is a different red marker. We can see that there's really only one spot for the red marker. So some red markers are made up of only one dye. We have this orange and this blue. We can see the blue traveled a bit further than the orange did. This orange is sort of a spear. You can look at how far your colors travel. Sometimes they'll separate out into multiple ones, like this one also separated out into multiple ones. Looks like I have some marker on my finger from doing this. So these separated out into some multiple ones. It's a little hard to see. It's a little faint right there. But there are some things that might go wrong. So what I have, let me show you. If you were to use something that isn't a washable marker, what will happen is it probably won't separate out very well. So it looks like this one was probably not a washable marker. This is my one. I did a couple of examples yesterday. This little spot right here, it was probably not a washable marker. Now, some of it seems to have traveled up a little bit. We can see there's still a dot down here. And the reason for that is if you use something like a Sharpie that's not a washable marker, so doesn't have chemical compounds similar to water, the water won't do anything to it. If you wanted to do a fun second experiment, you could use a Sharpie, but instead of water, you're going to want to use something similar to the dyes in the Sharpie. And in that case, if you have rubbing alcohol at home and you use that, if you use rubbing alcohol in here instead of water and you use a Sharpie, the Sharpie should separate out a bit. And then as one example, just for those who are going to try to follow after this, I said at the start that I was putting a bunch of dots. I was dotting it 10 or 11 times for each spot. If you don't put enough dye, this is what it will look like. Just so if you get yours that looks like this and you wonder what happened and why it doesn't look something like this or something like this, which is a bit more colorful. This I just didn't put enough dye on, so I didn't spot it enough. And so sort of the orange came up, the green came up, and I think I used purple over here. There just wasn't enough dye. So if you get something like this, it probably was that you just didn't use enough dye. So what you can do is once you're all done, you can put a little eye and a little smile and maybe some little antennas, and you have a very colorful caterpillar. Now it's our little chromatography caterpillar. So does anyone have any questions about the chromatography or the chromatography experiment, or even our fun trivia questions?
Go ahead and share in the comments if you guys have any questions. Tell us a little bit more. You mentioned that everyone's going to get that attended live is going to be getting something from your classes. So tell us a little bit about that. Where do they sign up to get that? Do they contact you via Facebook message?
Yeah, so you can either contact me via Facebook message, or if you go to hypatiastem.com, there is a link under the course catalog for our Science Sleuth Club. I also have, there is a list for the month of October. I tend to put them up every month. There is a list for the October dates in the experiment. So, if you've already done an experiment or you're not interested in one, you can look at what experiment is most interesting to you and choose the week for that one. And then if you're interested in a full, is it okay if I share my screen again? Sorry, I forgot I did have one more slide.
Yeah.
So in this case, I do also have, we do have a lot of classes. So if you're looking for sort of a full lab, this was sort of a fun experiment where we talked about chromatography, but if you want sort of a full lab where we talk more about capillary action and how it works and some different types of chromatography where we look at maybe some examples. I do teach middle school physical science and high school chemistry. For each of these classes, we have 2 classes per week. One is live and one is recorded. And each week we have a worksheet or a handout and readings to sort of back up what we're learning in class. And then throughout the semester, we also have 8 to 10 interactive labs depending on which class it is. So if you're interested in this, this is also on hypatiastem.com. We love doing our classes, and we sort of make sure that we have enough interactive hands-on things to really make sure that students are understanding our science concepts. So it looks like I thought I saw a question right before I shared, so let me come back over here. What colors are most likely multiple dyes? Oh, yes. So it depends on your brand. I know the black is, let me look at what I've done. I know the black is. I know the yellow is not. But it depends on your brand. So if you're using Crayola, I know the black is. But I have found, so I have these. I don't know what brand they are, but you might have to test it out. So I have these, and I know a couple of these are multiple colors as well. So different brands have different dyes essentially, and even they sometimes change their formulations. So certain Crayola reds are different than certain other reds. So if there's specialty packs, like the scented ones are slightly different, and things like that. So it'll really depend on what brand you use.
And you were mentioning that they were going to be able to get a free week of the Science Sleuth Club. Is that available right now for them to sign up
Yep.
for?
So you can either
Fabulous.
Facebook message me, sorry, or you can email me through, there's a contact form on Hypatia STEM, and just let me know you were in this class and if you want to, what week you would like to do. Our first one is DNA extraction. I think our second one is what I call monster Punnett squares, where we make some monsters with different features and we practice doing Punnett squares, which is sort of a genetics experiment. I think we have slime, and I want to say the last one is ice cream, but I might be mistaken because I know our last one in July was ice cream, and I don't know if I repeated it or not. I can't remember because I remember debating if I was going to repeat ice cream or not, and maybe I put a different one for the last week. And then, yeah, so it's open. Our first one, I believe, let me double check before I say the date. It's a Thursday. So it would be October 3rd, we start back up, and it'll continue all the way until early December. And then we have a break, and then it starts back up in about February. We go till May or June, have a break, and then continue on.
Thank you so much for doing this. This was a super fun class, and everyone had a lot of fun.
Well, I'm glad. And if anyone is doing this after the class and has any questions, feel free to reach out and I can help you troubleshoot. Or if you would like to share some pictures with me of your experiment, I would love to see them. Oh, yep, I can put in the chat. So it is, I'll just get the direct link really quick.
Yes, Jennifer, there is a website that you can go to. She's going to send you the direct link to the website where you can be able to see this schedule, and then you can pick the week that you would like, and then she'll get you set up with that.
Let's see, I put it in the private chat. I don't know how to put it in the, perfect, thank you so much.
I shared that.
Thank you
Okay,
so
so that's
much.
the direct link there in the comments. So you guys should be able to go there and see the class schedule and then contact Dr. K. Thank you so much for hosting this class. This was a lot of fun, and we look forward to seeing more from you in the group.
Thank you so much. Thank you so much to everyone who came. I had a great time.